Hello everyone, I'm the Father Rizky Fatullah GPM. I'm one of the author of our research that have topic the potential of the bromelain enzyme as the safe anthelmintic and minimum residues for the livestock commodities. With the member others are Dr. Yusuf Ridwan TVM MS and Dr. Rini Majestuti Vet Pharmacist. Our introduction of the research is Indonesia is one of the biggest country which have a potential livestock development for the society, economics, and the protein consume. In order that develop of the livestock, we must the good management and good health of the animal. One of the problem in our livestock is the infectious disease, such as the worm infection. Worm infection is, or in veterinarian we call it helminthiasis, is divided from the cystode, trematode, and nematode based on the classification of the worms. Classification difference worms based on a celome, like a anthelmintics, as salomata, and nemathelmintics, pseudosalomata. And one of the species of the group of the worm is the infectious disease for our livestock. And for the reduce the infection of worm, many veterinarians give anthelmintic drugs for the killed and reduce the infection. In order that many reports in the world that called anthelmintic resistant and anthelmintic residues problem, especially for animal production, with production from for meat, egg, and dairy milk. In order that we must get more the new candidate of drug for anthelmintic, and we search for the anthelmintic candidate from the nature, such as bromelain. Bromelain is the enzyme from the natural resource like a pineapple that have a potential active for anthelmintic, because bromelain has a proteolytic enzyme that a potential for kill the helminth, especially for reduce and broke of the integrity of the tegument and cuticle, and respond for muscularly, especially receptor in astekolin and acetylcholine esterase. Method of our research are: first, we must decision of the receptor of the target from its helminth infection in the livestock, and we divide it by antihelminthic and nematode. And second, we do of the molecular docking with USF Chimera. And the third, the last, our method is our prediction for toxicology and potential development in veterinarian. Result of and discussion of our research that are receptor of helminth, such as tubulin and acetylcholine, and we do the molecular docking for that with quality control of the drug are albendazole. The result are of the bromelain and albendazole. Bromelain have a bond interaction with the tegument and cuticular, especially in beta tubulin, but low bond reaction and interaction with acetylcholine than albendazole. And the toxicology and chemical profile of the bromelain are: bromelain have a good potential for anthelmintic drug because bromelain have a no for the absorption, and bromelain has a good excretion, and there are no potential for inhibition metabolism, especially CYP inhibitor in liver, and bromelain have potential of the gastrointestinal oral application. Our conclusion is bromelain enzyme is a proteolytic enzyme that have a good potential for oral anthelmintic for the next era, and bromelain have no risk for the anthelmintic residue because the bromelain have a good excretion metabolism, and bromelain is not toxic for any livestock animal, and bromelain is the one of the enzyme that's a natural enzyme can be absorbed and by the body, and bromelain have a no potential toxic for nervous central system. In order that bromelain have a good potential for anthelmintic in livestock for the new era. Thank you for our session.